flip for one, rice for two. I really like chocolate, so I wanted to talk about it specifically.
No, I did. Completely screwed. I put it in the dark hole trap. What you class. Trying to spring, so we'll see.
So
Good afternoon, everyone. Welcome to class. Let's go ahead and get started. I'd like to begin with prayer. If there's someone willing to give us a word of prayer, get us going. Please. Yeah. Just right there. Just
Our dear Heavenly Father, we're thankful that we can all gather together in person and virtually today to be able to study together about biochemistry. And we pray that as we do so, we might be led by the Spirit and be able to learn from these eternal principles and physical principles that govern our bodies and the world around us. We're grateful to be students here, and we say these things in the name of Jesus Christ. Amen.
Thank you. All right. Sorry. Just doesn't want to stay on. All right. So we are going to begin chapter 3 of Leninger. And some of the takeaways, some of the things that I would like you to pay attention to are the amino acids. So my expectation is that you will know the names and abbreviations for the 20 common amino acids. You do not need to know how to draw their structures. So that's not something you'll need to know, but you should know sort of whether they are polar, hydrophobic, aromatic, whether acidic or basic, just so you can classify them. But furthermore, there are 7 amino acids that have side groups, and we'll talk about what a side group is in a moment. But 7 of these that have ionizable side groups, that is, the side group acts as an acid or base. And consequently, it can lose or gain a proton, depending on whether it's an acid or base, respectively, and can be charged. Yes?
Do you know the 3-letter and the 1-letter code, or just?
Yeah, primarily the 3-letter abbreviations are fairly easy because in most cases they simply truncate the amino acid after 3 letters. The 1-letter ones are actually more complicated. But those are the ones you see in the literature. Okay. So if you're reading a medical journal article and they list out a new protein, it will all be with the 1-letter code. So you should pay attention to those. Also today, we're going to talk about something called the isoelectric point, the PI, which is it is the pH at which the charge on a peptide or protein falls to 0. And this has more significance in trying to work with proteins, trying to isolate, purify, and identify proteins than it does on their behavior in a physiological solution. Okay, so that's sort of where we're going today. I am going to try to move from here to a slide on PowerPoint, if I can do this. Okay, at the end, or towards the end of last class, I showed this slide and pose the question as to why the acidity or the relative acidity of the carboxyl group and the relative basicity of the amine group change as you link together multiple alanines. So let me walk you through this. Let me get to. So this particular pKa is for the acid, the carboxyl group that's on alanine. So alanine has a central carbon, and on one of its four constituents has a central carbon, and one of the four constituents is a carboxyl group, and it has a pKa, a nominal pKa of 2.34. It also has an amine group, and this is the pKa for that particular amine. All right. Now, if I couple together two alanines, I form a peptide bond between one copy of alanine and the second copy of alanine. I have now a dipeptide, but notice that the pKa for the carboxyl group is higher, which is an indication of weaker acidity. And likewise, the pKa of the amine group has fallen, indicating that it is now a weaker base, has less tendency to pick up a proton than a single copy of alanine. And the question is why? What do we have happening here that changes the pKa? Yes?
Would it be that the conjugate, I guess, base or the acid, it's more stabilized with more parts to it throughout the structure?
That's one way of expressing it. Work with me on this one. Any other ideas? Yes?
Well, so if the conjugate base was more stable, wouldn't that make it more acidic because it wants to go to that more?
Probably. So, yeah. Yes?
The addition of any group there that's a little bit electronegative at least will the electrons try and take the electrons away from the proton.
Okay, so the idea is that, if I'm hearing you correctly, proximity of a group that can withdraw an electron or withdraw a proton if it's closer, it's going to have a more powerful effect than if it's more remote. So let me get out of this and let me see if I can go back to where I was. Oh, okay. Okay, so let me see if I can. So if you can imagine now a tetrahedral carbon. So here we have we have a central carbon, we have two constituents. And one of these is a carboxyl group, one of these is a base. And they're fairly close in terms of physical space. Consequently, we have nitrogen. Okay. We have nitrogen with a pair of electrons. That's going to be relatively close to this carboxyl group. Now so that this pair of electrons, which is now physically close to this proton situated on the carboxyl group, is now influenced by this pair of electrons. In effect, that pair of electrons activates or promotes the loss of that hydrogen, that proton, And the consequence of that is that it is more acidic. And the same is true for the presence of that carboxyl group with its proton makes that amine group, the primary amine, more basic. Now notice what happens if I so if these are my acid and base groups, if I now insinuate space between them. I have a peptide bond and I have, so now my carboxyl group and my amine group are positioned at a greater distance one from another. And consequently, the acidity of the carboxyl group, the basicity of the amine are weaker. So what's the point in talking about this? The reason to discuss this is that, put another way, the microenvironment of these various functional groups on a protein matters enormously. And we will see situations where something that is considered under most circumstances to be inactive, relatively inert, can be activated and now becomes the species that attacks a substrate and initiates a reaction. This can happen because proteins fold. You have this long, you have an array of amino acids coupled together, but they fold. And as they fold, you bring together, you bring proximate one to another, these side groups. And as a consequence, you can dramatically affect their relative pKa's. You can make them much more basic or acidic, depending on what is neighboring that side. Okay, we'll see this again as we move forward. Okay, let's talk now about proteins. Okay, so what are some functions? Yes?
Can you quickly explain one more time that relationship between the lone pair and the hydrogen on the oxygen?
Okay, yeah, so if I can find this. So, okay, let me, if we have tetrahedral, sorry. So here's alanine, okay? Methyl group, central carbons, amino acid. Here's the acid functional group. Here's the base functional group. There are a lone pair of electrons on the nitrogen. This makes it a base. It is able to accept protons. That is, by definition, what constitutes a base for arrhenius acids and bases. And this is positioned now at fairly close to this hydrogen, which is attached to the hydroxyl of the carboxylate. And the consequence of having this pair of electrons next to this hydrogen means that this hydrogen will come off more easily as a proton, forming a bond to this pair of electrons. So we now protonate the amine to be an ammonium species, and we convert carboxylic acid to carboxylate, negatively charged. And that's accomplished because these are close. Now, if you position the carboxylate and the amine farther apart, it has less of a tendency for this proton to come off. Okay? Did that answer the question? Okay. All right, let's now talk about proteins. So what are some functions of proteins, quickly? Yes? Okay, enzymes are a very large class of proteins. These are able to facilitate, catalyze biochemical reactions. Okay, other things that we think about when we think about proteins. Yes?
Structure?
Okay, structural proteins. So this could be something like bone, actually, which has an organic protein core, and then it's mineralized. It could be something within the cell, such as a cytoskeleton, an actin chain, or series of chains. So yes, structure. Okay, something else that proteins do. Yes?
Transport.
Transport, okay. And here, there are molecules such as hemoglobin or myoglobin, which carry oxygen. There are other proteins in the circulation that allow for transport of selected materials through the bloodstream. So yes, transport is one, yes. Receptors, okay. So I'm just going to refer to this as signaling. So this represents molecules that, let's say, are regulatory compounds, hormones, cytokines, that work either systemically or locally, and they bind to receptors and activate processes within cells. So signaling, yes, yeah.
Would you put like motion?
Yeah, so motion is and this refers to everything from locomotion, that is my ability to walk across the room or to move my hand, down to things happening at the molecular level. So there are things that, there are some molecular motors that we will encounter as we go through the semester. One other major category. So the other is, I guess we'll just call it, well, let's, so aspects of the immune system, protective proteins, antibodies, immunoglobulins, and so forth, are proteins that serve a protective, defensive type of function within an organism. Okay. Now, the remarkable thing is that we're able to carry out extraordinarily different kinds of activity by simply assembling amino acids in a particular way. Okay? Same 20 amino acids, we simply piece them together differently in polymers. They're unbranched polymers, but this ability to do remarkably different things is due to now the side groups present on these amino acids. So when we talk about 20 different amino acids, we're talking about 20 different side groups. So let's consider that for a second. Let's see if I can get back to where I want to be. This is probably not where I'm supposed to go. All right, so just a little bit of intro. Are you seeing that? Okay. So proteins use the same 20 amino acids. There are a few exceptions, but the vast majority of proteins are assembled simply by the number and type of amino acids that are pieced together. They all have common names. Many of these common names arise from features of this, where they were found, some of their physical properties or other kinds of properties. So glycine is named because it tastes sweet. I don't recommend going around tasting things in the laboratory, but that is a feature of glycine. Asparagine was isolated from asparagus, and so hence we have it. All of these, at least all the ones that our body uses, with very, very few exceptions, are all L amino acids. So they have stereochemistry, all except for glycine. Okay, so this is the general structure of an amino acid. It has this central carbon, referred to as the alpha carbon. It has an amine group, and it has a carboxyl group, hence amino acid. And then it has this R group or side group, and this is what gives rise to different chemistry, different structures and different functions for proteins. As mentioned, these are all L, even though we show D-alanine here, at least in humans, there are, so far as I know, there really are not D amino acids. In some bacteria, you will find some D amino acids. This refers to a type of stereochemistry, D versus L, and this nomenclature was developed by a gentleman named Fischer. So these are Fischer structures for these. What you will see is that for the functional, so for L, the piece over here determines it's on the left side, levo, and over here it's on the right side, dextro, and so that is how these are named. But all you need to remember is we're talking about L amino acids. We won't really talk about any others. Okay, so here are the general categories of these amino acids. There are several of them that are nonpolar, and they are largely just hydrocarbon side chains, okay? So we start with glycine, which has just a hydrogen, alanine has a methyl group, and so forth. You can see that some of these are small, and some of these are quite large, but they share in common that they all have this nonpolar side chain. Proline is interesting. It is cyclized. That creates a shape to it that is very different. It's much less flexible than other amino acids. So this is one category. Then we have aromatic amino acids. So tyrosine, tryptophan, and phenylalanine all have aromatic side chains. And this gives them a slightly different chemical characteristic. They're also bulkier, as you can see, as well. Then we have polar compounds as side chains. So we see things with hydroxyl groups. So serine, threonine have a hydroxyl group. We have asparagine and glutamine, which have these amide features to them. So what's shown in the shaded area of the asparagine and glutamine are these amide groups that sort of terminate the side groups there. Those are polar. They're not ionizable. That is, you can't cause them to be charged. Cysteine is interesting. So it's a sulfhydryl group, and it has this SH group. It actually is ionizable. It is an extraordinarily weak acid. It can lose that hydrogen and become negatively charged. We then have basic amino acids. These all have amine functional groups on their side group. So we have lysine and arginine, which are strong bases, and we have histidine, which is a secondary, sort of a secondary amine. I say sort of because it is, there is delocalization of the electron density in in its side group here. So this side group here, this imidazole ring, is actually aromatic formally, and there is delocalization of those double bonds around the ring, and so it's somewhere between a secondary and a tertiary amine, which makes it weaker. So I don't know if you remember that from organic chemistry, but primary amines are stronger bases than secondary amines. And then finally, we have the two acidic acids, aspartic acid, glutamic acid, aspartate, glutamate. And so these two are going to be negatively charged in physiologic pHs, typically. Okay, so this represents them. So we have the nonpolar sort of aliphatic, we have the nonpolar aromatic, we have polar, but typically uncharged side groups, and then we have the basic ones, which are bases and can accept a proton and become positively charged. We have the acidic ones, which can lose a proton and become negatively charged. So these are the building blocks with which we have to work. Okay, now, this plot here shows you what the acid, so the pK1, if you look at the top here, this pK1 represents the acid of the amino acid, not the side group, just the acid functional group. The pK2 represents the base functional group here, but notice there's also a pKr, and this is an indication of whether the side group can be positively charged or negatively charged. If it's ionizable, if it has a pKa, then it can be charged. You'll see here that we have one in our aromatic amino acid. So we see this pKr for tyrosine of 10.07. Now, it's above 7. That doesn't make it a base. So pKa simply are an indication of how strong or weak an acid or base is. And in this case, it's an extremely weak acid. So tyrosine has a hydroxyl group. It actually has a phenol group. It's a benzene ring with a hydroxyl group attached. That hydroxyl group can, under certain circumstances, if you have a high enough pH, that proton will come off, leaving an O-. So it is an acid formally. Okay, and then the other 6 ionizable side groups are shown here. We have cysteine, which also acts as a base.
Hello.
And it can lose a proton and become S-. So it's an acid, it's a weak acid. You'd have to have a fairly high pH, or you'd have to have something else close by to activate it. We have our basic ones, and we have our acidic ones. Okay, are there questions about this? So I am asking you to know the pKrs, the pKas for the 7 ionizable side groups. You don't need to know the pKas for the acid group or the base group of the amino acids, but for the side group, these 7 do come into play and are important. Okay. Questions? All right, let's. Okay. So shown here is a hypothetical, sort of a theoretical titration curve for glycine. So its side group is just a hydrogen. It's not ionizable, so we only have the acid group and the base group, the amino acid part of it. that can be ionized. And what you're showing here is that we're effectively adding hydroxide. That is, we're increasing pH, and we're looking at the charge on this molecule as a function of time. And if you look at the very top, you'll see that there are various transformations that happen when we encounter a pKa. So you'll see the pK1 is 2.34. So we go from having a protonated carboxyl group, COOH, to a deprotonated carboxylate. So we go from neutral to negatively charged for our acid species. And that transition happens at a pH of 2.34, which is identical to the pKa, the pK1 of the acid group. At a pH of 2.34, half of the carboxyl groups will be protonated and half will be deprotonated. So it is a progressive process. So, okay. It gets more complicated with something like histidine, which does have an ionizable side group. So again, if you look at the very top, you can follow what's happening as we increase the pH surrounding the histidine molecule. So we go from everything being protonated. So the carboxyl group is protonated and neutral. The amine group is protonated and positive. And the ring, the imidazole ring of histidine, is also protonated at a low pH and also positively charged. So we have a net charge of +2 on this. As we encounter now, as we raise the pH, we begin to lose the proton off of the carboxyl group. And we begin at a pH of 1.82, we have half of the carboxyl group deprotonated and half protonated. Hence, on average, we have a net charge of -0.5. So we've gone from a net charge of +2 to 1.5. As we move beyond the 1.82, that carboxyl will become fully deprotonated and be negatively charged. Then we encounter, as we go up in pH, you'll see that as we go up, we are going to then we're going to now deprotonate or begin to deprotonate this ring here, and we will transition at a pH of 6 to a deprotonated neutral ring. So this is a weak base. Consequently, it doesn't take much of a increase in pH to remove that proton. Okay, and then finally, if we get the pH high enough, everything will be deprotonated. That results in a carboxyl. You get rid of the, is there a way to get rid of the banner up there? Okay. Apologize. You can't see the carboxyl group up there. Okay. All right. Okay. I'm sorry. Okay. So as we go to a higher pH now, up to, so notice that the final pKa, is 9.17. Above that, then everything becomes deprotonated. Our carboxyl group is deprotonated. This amine is deprotonated, it now becomes neutral. The ring was deprotonated at a lower pH and is neutral. Hence, the net charge on this molecule is a -1. Okay. Okay, so here's the question for us. The interior of the cell is buffered. That is to say, you have a relatively fixed pH. So, which amino acid side group would you, that is part of a protein, would be likely most sensitive to the cell environment or the pH in the blood? Is there an amino acid that has a side group with a pKa that is sort of in the physiologic pH range? Yes? Yeah, histidine. Okay, so that ring on there. So actually histidine becomes, in some ways, a very important player for something that you can change fairly easily by adjusting pH, and this allows for it to participate in a number of different processes. We will see this extensively in hemoglobin because there are a number of histidines that actually lead to changes in oxygen binding affinity. So other amino acids can participate, actually initiate chemical reactions. So this means, let's say, initiating the conversion of substrate to product for an enzyme. How is that possible? Let's go back and think about that, the alanine and polyalanine, and remember that the microenvironment, what's next to a functional group can either activate or inactivate that side group in a folded protein. You bring things next to it, and that can alter its effective pKa. All right, very quickly, we're going to talk about the formation now of peptide bonds. So this is a synthetic process. Put another way, thermodynamically, it is not favored to form arrays of amino acids. It's not favored energetically to form proteins. So the reactants are more stable than the products. Nevertheless, once you form the product, once you form peptide bonds, they are actually quite stable. To digest a protein, just to put it in perspective, when you digest proteins all the way to amino acids, it requires 6 normal HCl, and you boil them for 2 hours to be able to break apart the peptide bonds. So quite stable once they are formed, but nevertheless, the reactants are more stable than products. Okay. So here we go. This represents now the formation of a peptide bond. It is an amide bond. So the organic chemical term for the coupling here is an amide bond. The C double is carbonyl coupled to this nitrogen is an amide bond. But it is also, in the context of biology, referred to as a peptide bond. Let me note that while the initial carboxyl group here had a pKa, that is, it could lose that proton, and this amine here was quite basic and could gain a proton, there is no pKa associated with the peptide bond. It is not ionizable. So that changes things for us. Okay. When we look at these peptides, we consider them from the N-terminus, that is, we consider the free amine that is present on the left side as shown here. We talk about this being the N-terminus, the amine terminus, and we talk about the C-terminus, or the carboxylate group on the other end. So by nomenclature, we name things, or we talk about the sequence from N-terminus to C-terminus. And that's the convention that's used for full-length proteins as well. Now, here you see that we can have side groups that also have charge on it. We have a charge on the N-terminus, typically. That amine, unless it's been modified chemically, will be protonated. We have a C-terminus with a C double bond, CO2 minus, but we can also have side groups. So we have a glutamate here, right here, and we have a lysine with a free amine here, and those can be charged they can be charged depending on what the pH is. Okay, we are now going to spend a few minutes and learn to how to calculate the isoelectric point of a peptide. Okay, so here's my peptide. It's got a leucine. Sorry, let me get out of here so I can go back to. Okay. So. Okay, so this L represents an leucine, and this has this amine group on it. Okay. I have, let's say, an alanine, and then I have a histidine, and I have a cysteine, a threonine, another alanine, and at this end, I have a carboxyl group. Okay. Now, these dashes between these letters represent peptide bonds. So when we're talking about calculation of isoelectric point, we're talking about those species that can bear a charge. Amines are bases, and they can be positively charged. Carboxyl groups can become carboxylates and be negatively charged. Our histidine has this ring system, this imidazole ring, and over and our cysteine has a sulfhydryl group. The sulfhydryl group acts as an acid. It can either be SH, or if the pH is sufficiently high, it can be an S minus. It can't be positively charged. Hence, it is acidic, even though extremely weakly acidic. Okay, are you with me so far? Questions about this? So I'm setting this up. I just, over here, this has a pKa, so let's just list these out. This has a pKa of 2.34. This group over here, 9.60. This imidazole ring, 6.0. And our sulfhydryl group here, our cysteine, 8.18. So this is basic, not because it's high, but because it's an amine and can accept a proton. This is basic, even though its pKa is below 7, because it can accept a proton and become positively charged. The sulfhydryl group, despite having a pKa of 8 plus, can lose this hydrogen to become negatively charged, and this is acidic. We can lose a proton and become negatively charged. Now, what I'm going to do is carry out a virtual titration. So if you'll bear with me, I'm going to switch boards here. And to do this, I'm going to use this kind of a bookkeeping method to keep track of those 4 ionizable groups. So I'm going to list them out. So here we have, here I'm going to change the pH. So the pH is going to change. Down here are the pKa's of those four groups. So we have the carboxyl group. Okay, so this is this. I have the imidazole group. I'm just listing these in order of pKa. And I have this ring structure. Oops. And I have a cysteine, which this, 8.18, and finally I have the amine group on the N-terminus, 9.6. Okay, now I'm going to begin with a pH that's below the least of these pKa's. So at a pH less than 2.34, everything will be protonated. So what that means is this carboxyl group will have the hydrogen in place. It will have no charge. This will be protonated and positively charged. This will be +1. This sulfide group will be protonated and neutral. This will be protonated and be a +1. Okay, have I lost you? Go ahead and ask questions. We'll take it. Okay, the next point we have to worry about is where this, the pH, I'm artificially increasing the pH. I raise it now to exactly 2.34. Why? Well, that's because that is the pKa of this first group that we have to worry about. And at that point, this proton begins to come off, leaving us with a CO2 minus. But at 2.34, on average, there is a mix. Some of them have their proton on, some of them have the proton off. So the net charge is a -0.5. Notice that the 2.34 is well below 6.0, 8.128, and 9.6. So these will continue to be protonated. So here we have a net charge of +2. At this point, we have a net charge of 1.5. Okay, now I'm going to consider the interval. Yes?
This is a question about the half charge. So at 2.35, is it like 70%, or is it like a threshold drop at 2.35?
So at 2.34, at a pH of 2.34, it is really in equilibrium. So half will be protonated, half deprotonated. Okay, so the net is a -0.5.
But at 2.35, like you go put one.
No, it's progressive. It's not
So
precipitous.
it's like 70% to 90%.
Yeah, I mean, the log scale, it is a log scale, so it does change more rapidly than you might anticipate. So, okay, now we go between, now we go between 2.3 and up to all the way up to 6. So now we allow the pH to rise. In this region, this becomes fully deprotonated, but we're still, we're below 6.0. So this is above 2.3, below 6.0. This will still be predominantly protonated. We're well below the pKa here and here. So these remain unchanged. Now we have a net charge of 1.0. Okay, and then we encounter a P. Okay, and then we now actually move to a pH of 6.0. We're well above the pK of this, so this will remain deprotonated. At 6.0, now we begin to establish this equilibrium between the protonated and deprotonated form of histidine. So we now have a net charge of 0.5. These remain unchanged because we are still well below the pKa's available. Okay. Now the net charge on this is just 0.5. Then finally, well, not finally, but now we'll consider the interval between above 6.0 but below 8.18. And in this region, we end up with no net charge. This is then the isoelectric point. So halfway between 6 and halfway between 8.18, in the middle of that interval, we will achieve chemical neutrality. We will have as many negative charges as positive charges. And so to calculate the isoelectric point, we take these two, we just average them. We just find the midpoint. And this is going to be 7.09. So the isoelectric point, at which charge falls to 0, is 7.09 for this peptide. Yes?
So this is going to have a value.
Not as yet, okay? But this can allow us to isolate proteins based on their chemical characteristics, okay? We'll see how this happens next lecture when we talk about separation methods. There are ways of doing this so that if it's positively charged, we can pull it out one way. If it's neutral, we can do something else to try to capture it and so forth. All right, I apologize, no story today. We're past time. Thank you for your patience. Apologize for the backbeat on the sound.